Hello, my name is Koen Gistelinck and I will be presenting the importance of light microscopy in my PhD research, with a focus on the study of promoters and gene expression. First, some background concerning the topic. Lectins are a class of proteins that have the ability to recognize and bind carbohydrates in a reversible way without modifying these carbohydrates. You can see these proteins as tape. They stick to sugar. On the other hand, glycosyl hydrolases are enzymes that cleave glycosidic bonds, resulting in the release of a carbohydrate. These proteins can be compared to a scissor. Lectins and glycosyl hydrolases both have the same interacting partner, namely carbohydrates. You can ask yourself whether or not these two types of protein occur together like a hybrid tape-scissor. The answer to that question is yes. These hybrid sequences are present in several plant genomes. So, my goal is to unravel the functionality of this type of hybrid proteins. I'm trying to find the answers to the following questions. What do the proteins do? To study the proteins, recombinant protein is made and subsequent biochemical assays are being performed. Where is the protein present in the cell? I want to know because a given biochemical or biological function is closely related to the location of the protein. And lastly, when is the gene of interest expressed? Does the expression of the gene occur during a particular developmental stage, or is the expression induced by an abiotic or biotic stress? To be able to answer all these questions, a combination of microscopy and molecular techniques can be helpful. One of the instruments that can be used is a confocal fluorescence microscope. The figure explains how this technique works. Generally, fluorescence requires a fluorescent molecule, a laser that will excite the molecule, a detector that intercepts the fluorescence light, also known as emission, and a dichroic mirror that reflects the excitation light but allows the fluorescent signal to pass. The issue with fluorescence microscopy is that the complete sample is illuminated, and the fluorescent signal from both above and below the focus plane are detected. This results in a lot of background signal. In confocal microscopy, the excitation light is instead focused in a specific spot in the sample, the so-called focal point. The use of a pinhole before the detector allows to block the out-of-focus light, so only the fluorescent signal in the focus plane will be detected. By scanning the focal spots, a fluorescence image can be reconstructed. To visualize whether the protein is present in the cell, the protein of interest was linked to a fluorescent molecule. Here, green fluorescent protein, or GFP, was used as a marker. GFP was introduced in plant cell cultures or tobacco leaves and can be detected through confocal fluorescence microscopy. The following slides will display some of the results. This slide displays an experiment where the protein of interest was produced in a plant cell suspension culture. On the left picture, untransformed cells can be seen. Very little fluorescent signal is visible. On the right side, the transformed cells are displayed. Fluorescent signal is observed, most likely in the endoplasmatic reticulum and the Golgi surrounding the nucleus, as indicated by the blue arrows. Additionally, bright green particles corresponding to transport vesicles are clear. These data suggest that the protein of interest is expressed properly. Here we see the GFP fusion protein in transformed plant cell suspensions. On the left, you can find the negative control. On the right picture displays the cells transformed with the GFP fusion construct. It is clear that the vacuole is stained nicely, but also the intersections between the cells have a bright signal, as indicated by the yellow arrows. The latter localization could either correspond to the cytoplasm, cell membrane, or the cell wall. To be 100% sure, further experiments are required, for instance using specific stains, for some of these cell structures. On this slide, the GFP fusion protein can be observed in tobacco leaf epidermal cells. On the left, you can find the negative control. There is little fluorescent signal visible in the untransformed cells. In the middle, you can see the typical shape of tobacco cells. You can find cells expressing the free EGFP to the nucleus indicated by the blue arrows and the cytoplasm indicated by the red arrows. The third picture displays cells that were transformed with the construct of interest. The fluorescent signal is present in the cell surroundings, most likely cell membrane or the cell wall. To answer the question when the proteins are expressed, another molecular tool can be used. The promoter of the gene of interest is placed in front of a reporter gene. In our case, this reporter gene is beta-glucuronidase, or GUS. This enzyme creates a blue precipitate with a substrate called X-glut. The promoter GUS constructs were introduced in Arabidopsis plants. Next, transformed plants were selected for GUS staining experiments. Light microscopy allows us to check in which tissues, for instance flowers, roots or leaves, the blue precipitate occurs. It is very important to include controls during these type of experiments. As you can see, the wild-type flowers of an Arabidopsis plant show little to no blue color. From this slide, it is clear that there is a blue deposition in the young stages of the flower. In later stages, only the pistil is colored, and in more mature stages of the flower, only the tissues surrounding the ovules are colored blue. This suggests a specific role of the proteins in flower development. Based on the result presented during this presentation, I hope I have been able to convince you that microscopy is an indispensable tool for my plant research. It helped me to answer whether the protein of interest is produced, where the protein is localized, and during which stages of plant development the promoter is active. A downside we could encounter for fluorescence microscopy is the presence of background signal or autofluorescence from the chloroplasts. Another issue is that the resolution of the microscope is insufficient to distinguish between, for instance, the cytoplasm, cell membrane or cell wall. But we have tools to help us solve this problem. And with these statements, I will conclude my presentation. I want to thank the organizers for giving me the opportunity to present my work, the Ghent Light Microscopy Core for allowing me to use the confocal fluorescence microscope, and my colleagues and supervisor for the support. Thank you for your attention.